Welcome to the next session on the chapter Respiration in Plants. In this module, we are going to learn about the electron transport system, which helps to release energy which is stored in the reduced coenzymes, that is NAD plus and FADH2, which has been formed during glycolysis and Krebs cycle. Okay, so glycolysis and Krebs cycle has yielded many NADH and FADH2. We know the total number of NADH produced as a result of Krebs cycle is 8. Okay, we had learned in the last module it is 8 NADH and 2 has already been harvested or produced during glycolysis when 3-phosphoglyceraldehyde is getting converted to 1,3-bisphosphoglycerate, and during that process 2 molecules of NAD has been reduced to NADH plus H plus. So total there are 10 NADH, okay, NADH, and there are 2 FADH2. So these reduced coenzymes is having a large amount of energy, and the energy present in these reduced coenzymes has to be released for the synthesis of ATP. So for that, it has to be passed to the next crucial event called as the electron transport system. Okay, so electron transport system is a metabolic pathway which is taking place in the inner mitochondrial membrane. When the Krebs cycle was taking place in the matrix, here electron transport system is going to take place in the inner mitochondrial membrane, and during this process electron passes from one electron carrier to the other, and this is done to release the your energy stored in NADH plus H plus and FADH2, which is formed during glycolysis and TCA cycle by oxidation. That is, NADH gets oxidized to NAD plus and FADH2 gets oxidized to FAD. And ultimately, at last, the electrons are passed on to the oxygen, okay, oxygen molecule to yield water. Okay, so we know what are the byproducts of respiration. It is carbon dioxide that we have seen in the Krebs cycle, which was forming Krebs cycle, and water. Okay, water was also released during Krebs cycle, and also it is going to be released over here. Okay, so and at last energy has also to be synthesized. That is a main aim of respiration. So let us see what are the electron carriers. Okay, so electron carriers, let us see the electron carriers over here. This figure properly we can see the electron carriers. This is the first complex. Okay, it is situated in the inner mitochondrial membrane. Okay, so this is the inner mitochondrial membrane, this is the outer mitochondrial membrane, and in between there is a space, and this region is the matrix in which the Krebs cycle has taken place. The first complex, complex 1, is the NADH dehydrogenase. Okay, it's called as a NADH dehydrogenase. We can see over here. It is the NADH dehydrogenase, the first complex. And next complex is the ubiquinone. It's an electron carrier, and ubiquinone is designated as complex Q. It is also the next followed by ubiquinone, that is complex 2, which is nothing but the succinate dehydrogenase. Okay, succinate dehydrogenase. And complex 3 is the cytochrome BC1. And the fourth complex is cytochrome C oxidase, which is connected together by a cytochrome C. Okay, so these are the different electron carriers which is present on the inner mitochondrial membrane. Okay, which is present on the inner mitochondrial membrane. So first, which is the complex called as NADH dehydrogenase, which is called as a complex 1. The function of this complex is to oxidize NADH plus H plus to NAD. Okay, you can see it is getting NADH is getting oxidized to NAD, and the function is done by the first complex, that is NADH dehydrogenase. When NADH plus H plus is oxidized to NAD, protons gets transferred from the matrix to the inner mitochondrial space. Okay, so the electrons is then transferred to the next carrier, which is called as a ubiquinone or the complex Q, which is located on the inner membrane. And the ubiquinone, along with the electrons received from NADH dehydrogenase, it also receives electrons from the second complex, which is called as a succinate dehydrogenase. This is a second complex called as succinate dehydrogenase. And the function of succinate dehydrogenase is to oxidize the FADH2 formed during the TCA cycle. So FADH2 is oxidized to FAD. And during this oxidation, it transfers the electrons, okay, to ubiquinone. So ubiquinone is receiving electrons from both complex 1 and complex 2, okay. And this ubiquinone, the reduced, when it is receiving an electron, it gets reduced, okay. And then when it is getting oxidized, it transfers the protons from the matrix, okay. It transfers the protons to the inner mitochondrial space. And this reduced ubiquinone, it passes its electrons to the next electron carrier, and this next electron carrier is called as cytochrome BC1. So this is cytochrome BC1, okay. This is cytochrome BC1, and the cytochrome BC1 gets reduced when it is receiving an electron. And the cytochrome BC1, it transfers its electrons to the last complex, that is complex 4, which is called as cytochrome C oxidase. And there's an electron carrier between complex 3 and complex 4. It is nothing but the cytochrome C. Here we can see the cytochrome C over here. And cytochrome C, which is carrying the electrons from cytochrome BC1 complex to cytochrome C oxidase, okay. And this cytochrome C oxidase, it is containing cytochrome A and A3 and 2 copper centers. So all these NADH dehydrogenase, ubiquinone, then cytochrome BC1, succinate dehydrogenase, then cytochrome C oxidase, all these are electron carriers. Electrons are being carried, okay. When are these electrons released? When NADH is getting oxidized to NAD and FADH2 is getting again oxidized to FAD. Okay. So when electrons pass from one carrier to another, from complex 1 to 4, they are coupled to synthesis. They are coupled to ATP synthase, which is complex 5. This is a complex 5, okay. Complex 5 is called as ATP synthase. So the electrons ultimately, okay, they are these 4 complexes are again, they are coupled with the 5th complex called as ATP synthase, okay. And it is actually the 5th complex, that is ATP synthase, which is involved in ATP synthesis, okay. And the number of ATP molecules, it depends upon the electron donor. If the oxidation of NADH is taking place, one molecule of NADH will yield 3 ATP. Okay, so one molecule of ATP will yield 3 ATP, and oxidation of one molecule of AD and FADH2 will yield 2 ATP. So the number of ATP synthesized will depend upon the electron donor. Here we can see the transfer of electrons from complex 1, that is NADH dehydrogenase, the complex 2, the succinate dehydrogenase via the ubiquinone complex, which transfers the electron to the 3rd complex, cytochrome BC1, via the cytochrome C, ultimately reaches the complex 5, that is cytochrome C oxidase. Actually, the role of oxygen just comes in. Okay, when it is a final acceptor of hydrogen, is removing the hydrogen from the system, and thus oxygen acts as a final hydrogen acceptor. And energy, in respiration, energy of oxidation reduction is utilized for the phosphorylation. We know each complex gets simultaneously reduced and oxidized. So this brings about phosphorylation. Okay, so phosphorylation means synthesis of ATP. So energy released from the ETS is utilized to synthesis ATP by the 5th complex called as ATP synthase. Okay, so ATP synthase has 2 major components. It is having F1 head piece and F0. What is this F1? It is called as a peripheral membrane protein complex, and F0 is the integral membrane protein complex. Here we can see the F0 is embedded within the inner mitochondrial membrane, and F1 projects into the matrix part. Okay. The passage of protons is coupled to the catalytic synthesis of F1 component, okay, for ATP production. So for each ATP produced, 2 protons passes from F0 via the F1 into the matrix, okay, from the inner mitochondrial space to the matrix, down the electrochemical proton gradient in order to break down the proton gradient which has been greater in the inner mitochondrial space. So when it is getting broken down, when this gradient is being broken down, it releases energy, and this energy is utilized by the ATP synthase to synthesis ATP. So that is the ultimate aim, to synthesis ATP from ADP plus inorganic phosphate. So that was the ultimate aim of cellular respiration, to yield energy. So it was a stepwise breaking down of the respiratory substrate to ultimately release energy, okay, through glycolysis, Krebs cycle, and electron transport system.